Hello, everyone, and welcome to today's Bite-Size Talk. I'm super happy that today we have James with us from the Max Planck Institute for Evolutionary Anthropology, and he is going to talk about a new pipeline that has been released recently, which is NF-core createtaxdb. And this is your platform now.
Thank you, Fran. So yeah, I'm talking about createtaxdb, which is a pipeline aimed at people primarily working in metagenomics. And if you're not familiar, what metagenomics is essentially the study of all of the DNA in a given typically bulk or environmental sample. So you're not working with isolates and analyzing one particular species. You're looking at the DNA of all the organisms that are living in a particular sample at any one point. And in any metagenomic study, pretty much always the very first question is simply just who is there? So where has the DNA come from in terms of the organisms who've donated it? And typically in metagenomic studies, the first way we do this is with an approach called metagenomic taxonomic classification or read binning or things like this. And what this simply means is you have all of your DNA sequences or molecules. You can't see my cursor, one moment. You have all of the different DNA molecules or reads, all from the different organisms indicated here in the different colors. And then we sort them out to basically find what is the most close reference genome that the DNA sequence matches. And we make the assumption then is that that DNA sequence most likely came from the organism that has that genome. So it's basically a sorting problem. So even though it sounds like a very simple thing, it's an unresolved problem. Taxonomic classification does not have one gold standard tool or collection of tools. In another organization or community I'm part in called SPAM, we've actually identified more than 169 classification software that have been published since 2007. So 169 different tools, all basically tackling essentially the same problem. Which means how do you actually pick each one? So this is from the CAMI challenge, these results. The CAMI challenge was basically a benchmarking project, which basically asked different researchers and developers to submit their tools with different parameters and run against synthetic and real data sets to see which one performed better in a variety of metagenomic tasks, one being read classification. And what you can see here from the very high diversity of names and the very inconsistent shapes of all of the lines here, whoopsie, sorry, is that there is never really one tool that basically outperforms the other. It ultimately depends on which context you need to pick your classifier for. So if you've got one context, one question, you typically have to use this profiling tool or classification tool. If you have another one, it'll be this one. But the biggest challenge is that we're dealing with metagenomics, and often we are dealing with multiple contexts. So a very common one is you may be interested in identifying both bacteria and viruses in the same sample, but different tools will perform differently and perform better on bacteria, and others will perform better on viruses. So how do you handle this? And ultimately, the best way is to run multiple tools. And the good news is that in NF-core, we already have a pipeline, which we probably, I think, 2 years ago presented in a previous bite-sized talk called NF-core taxprofiler. And what this does is you take your input FASTQ files from your metagenomic sequencing, run it through some pre-processing steps. But the trick with taxprofiler is it allows you to run multiple taxonomic classification tools on the same data set in one go, basically in parallel. So it means that you can pick and choose which tools you want to run, depending on the question you have for that particular data set, or questions, more importantly. So that's great. Uses Nextflow's great parallelization and things like this in a very efficient manner. But the eagle-eyed of you in the audience or watching this video may have noticed that compared to our normal metro maps, that we have something funky going on here, where we have 3 lines in a sort of gradient color for each of the classifiers. And this is all coming out of the databases. Now, the reason why this is special is that another component of the unsolved problem of taxonomic classification is the other biggest questions that we have is what database do you use? Because again, depending on the context, depending on what your question is, you may have to use different databases. And the problem here is that there is no gold standard database or sets of databases that you can pick. Uh, Compared to, let's say, reference genomes, where often you go to NCBI and they all have flagged probably the most complete, most polished, most well annotated genome that can act as the reference sequence of that particular organism. The same thing does not exist when it comes to metagenomic classification. And in metagenomics, there is actually a common misconception, even with people working in this field, that you can just use NCBI nucleotide or NT or RefSeq databases. But this actually has a lot of problems with it. If you were to try and index the entirety of RefSeq or NT, you result in gigantic databases which are terabytes in size. So in fact, the last I heard is about 2 years ago, if you tried to index NCBI nucleotide, you'd require about a 4 terabyte RAM node on your HPC, depending on what classifier tool you want to use. Also, much of there are many redundant sequences in these databases. So, for example, which results in a huge taxon bias or skew. So, for example, this is actually a little bit of code I did for a review that I was doing for a journal article where the authors had not actually described what was in their database, and I had to demonstrate that, look, you could be getting massively, wildly different results because there are 41,000 E. coli genomes in RefSeq, but there's only one Mycobacterium leprae genome. And what this means is that when you're doing metagenomic classification, most of these tools, if a read hits multiple reference genomes, it'll basically push that classification up the taxonomic tree that represents the organisms in your database to say, okay, we may have some Escherichia or something like this. So you don't get a precise identification just because you have way too many E. coli genomes that it's not balanced. Whereas if you have something else which is missing in your database, but it's something very vaguely related to Mycobacterium leprae, you may start getting hits to this pathogen, and you may end up false reporting the presence of this pathogen. Furthermore, there's other issues with database building, is that even with an NCBI-NT and particularly RefSeq, and RefSeq is meant to be a more cleaned up database, is not all genomes are clean. You will find genomes which have cross-contamination from other organisms. You'll have sequences with adapter sequences and other lab artifacts in them. So you can't just willy-nilly download these because you may be, causing other problems with these artifacts in these genomes. And also, NT and RefSeq, while they are some of the biggest or most comprehensive databases, they're not completely comprehensive. So another shift we're seeing now in metagenomics is a shift towards MAGs, or metagenomically assembled genomes, which are, rather than taking isolates, so single cells, and then basically sequencing those genomes without any other sort of organism there, we try to infer genomes from metagenomic samples when you have all these organisms using fancy computational methods, resulting in hundreds and thousands of very difficult genomes of very difficult to isolate organisms. And there are other databases such as the GTDB, which basically include these here, but they are not included in NT or RefSeq, even though many of these genomes are actually very, very important. So ultimately, because of all of these issues, which is the problem statement, is that you really need to build custom, thoughtfully considered databases for your taxonomic classification. So how do you go about doing this? So the considerations you need to make when building a custom database is, firstly, is the genomes you've picked, meaning you have a comprehensive enough database for your question. So i.e., does it include everything you're looking for, or things at least similar enough to match the things you're looking for? You also need to consider, what are the gaps in your database. So have you included necessary diversity around your targets? What I mean by that is there are some pathogens where actually they have lots of commensal relatives, so non-pathogenic, don't hurt you at all, but a very, very similar genome. The only thing that distinguishes between the two is the presence of a particular virulent plasmid or something like that. So to make sure you don't, if you're doing clinical metagenomics, for example, you don't want to misclassify a pathogen, you need to make sure you have diversity of the commensal relatives in your database around with the pathogen. Another thing is, after building, is your database small enough that it can fit into your infrastructure? This is a very common problem in metagenomics, particularly with the students in the different labs I've been talking to, is that they often ask, oh, okay, James, how do I make a genome that I can fit onto my 256 gigabyte RAM node, which is the largest one I have? If you just download the entirety of RefSeq, it's not going to fit. But taking into consideration, if you remove stuff, is your database still comprehensive enough? Then also, we are living in an era of a data deluge where we're producing probably hundreds or even thousands of genomes a month. So you need to have a way to basically update this regularly. Is your database clean? So as I mentioned previously, have you made sure that your genomes are not contaminated with adapted sequences or chimeric sequence assemblies? And also, what classifier do you want to use? Like I said before, you may, in many questions, have to run multiple classifiers, which are addressing different questions or targeting different species. So you need to consider, okay, what multiple classifiers do I need to build a database for when you're making your selection? My main point here is that there is not one size fits all database. You will, I at least would recommend that you should always basically think carefully and try and build your own. If there is no accepted form of database available, which generally there is not in the field. So that led me to the question, okay, what can NF-core do about this? And this is where the pipeline comes in, which is called NF-core createtaxdb. The pipeline takes in your FASTA files, whether that's nucleotide or amino acid, depending on the classifier you want to do, and taxonomy information. The FASTA files go through very basic cleanup here, and then you can build in parallel many different types of classifiers, databases for different classifiers. Then we can package it up in different ways, and then you get the databases themselves, compressed versions in cases where they have multiple files, and also TSV, which is the input sheet for NF-core taxprofiler. So hopefully that caught everybody up. Then I went on to the to-do slide. Hopefully you can see this.
Yes, we do.
Cool. So what's next? Basically, make sure that the pipeline stays in sync with NF-core taxprofiler. So we will be adding to taxprofiler multiple new classification tools, particularly in long reads. And the idea is every time we add a new classifier to taxprofiler, we'll also add a new database building module to createtaxdb. Talking about the other problems in terms of database sizes, trying to reduce this, I want to see if we can add different strategies for database reduction. So for example, filtering out or collapsing redundant genomes, whether that's via just using taxonomy information or actually clustering genomes based on ANI, so average nucleotide identity sequence scores, and picking representative from those clusters. It would be really nice to add contamination detection, so ability to flag any genomes which may potentially have this chimeric assembly or with adapter sequences in it. And in the long run, which I think will be quite nice, is maybe we could sort of move towards having a certain level of gold standard databases and basically offering that via database hosting, where we can regularly basically regenerate these as new genomes come in with the same sort of rough collection of genomes or taxa to act in a similar way to iGenomes as we have for NF-core or sort of genomic-based pipelines. So before I wrap up, I just want to do a little bit of advertising. As a part of the meta-omics special interest group and also the NFDI for microbiota, I'm planning to host a exhibition day, community exhibition day for NX for pipelines, whether that's metagenomics, meta-taxonomics, meta-proteomics, etc. Where the idea is that developers will offer short, maybe 2-hour sort of sessions where they give a little introductory talk, and then a hands-on demo of the pipeline, allowing attendees to actually sort of ask in real-time questions with the developer. And I'm looking for people or developers who are developing pipelines in these areas currently who would maybe be willing to give a demo session of their pipeline. It does not need to be NF-core, just anything in Nextflow is fine. So if you're interested, I would love to hear from you, so please register your interest with the QR code and the link below, or the link, I should say. And to wrap up, So thank you very much to Sam and Alexander, who were the main sort of co-developers of the pipeline. Also Sophia and Lily and Moritz, who I missed off the slide here, sorry, who helped co-design the pipeline really by our decisions made in taxprofiler. Paul for already adding useful documentation, who's using the pipeline for different things, and also Zandra for the logo, and of course always the NFPOL community as a whole. So thank you very much. Sorry about the technical difficulties with the slides, but are there any questions?
Thank you very much. And I see you get a lot of love already. So are there any questions from the audience? You can also unmute yourself now. So it seems people are still thinking. I was wondering, would it make any sense to have pan-genomes as input for your taxprofiler database creation, or is that something that is way too far?
That's a very, very good question. And I think that is the direction many areas of genomics will be going in, including metagenomics. So I'd say currently the tools that we support, and generally in metagenomics that I know of, don't really support pangenomic information. The data constructs would not really work in that you need for pangenomes would not really fit in the way that these databases are built, which basically require a basic FASTA file. But certainly in the long run, that will make these things much, much more efficient, become much more accurate in how you pick up, even strain level variation. So that once the classification tools start adopting this is probably
Yeah,
where we'll go. That's a very good question.
cool. Are there any more questions?
I recognize half the people anyway, so I know
Yeah, I have a
if I ask a
very
question, sure.
non-scientific question. I always
Yep.
wondered why you have the dachshund in your logo.
The dachshund is in the logo because Zandra, who designed it, is a Swedish-speaking Finn, and we share a love of dogs. And dogs are very good at identifying microbes. And conveniently, tax is the Swedish word for dachshund. So
Ah,
taxprofiler, createtaxdb, dachshund, dog.
nice. Thank you. So last chance for anyone to put in another question. Otherwise, I thank you, James, for this
Thank
very
you, Fran.
informative talk and everyone for listening in. And I hope to see you in our next bite-sized talk. Have a great day.
Bye.
Bye bye.